Sure. All right.
Are we ready, Andy? Okay. So everyone, welcome. Thanks for braving the snow and cold to come out for the first Wednesdays at NICO talk of the quarter. You can see up here all of the excellent speakers that are slated for the coming, I guess, 8 weeks. And but today I'm glad to welcome István Kovács, who is our colleague here at Northwestern in the Department of Physics and Astronomy. I just want to make this a quick intro. You should talk to him, get to know him. He's around here, so if you want to know more. But just a couple of highlights. István's from Hungary, and he excelled there, and then came and did a postdoc in the U.S. at Northeastern University with László Barabashi. But I also learned that he was at the Dana-Farber Institute at the same time. So he's got experience both in the medical school side and in physics. And since he came to Northwestern, he's been doing a lot of amazing stuff. His research covers a broad range of fields, very appropriate for NICO, from quantum physics to biophysics, I think maybe more what we'll hear about today, and complex networks, to also things like the magnetization, the Ising model, and critical phenomena, phase transitions. So in addition to doing all this amazing research, he's also somehow won an award for improving undergraduate experience at Northwestern, 2 mentoring awards also for undergraduate research. And most recently, I saw he won an NSF Career Award, which is an amazing accomplishment, big competitive award for early career faculty. So congratulations on that, István. And I'm looking forward to hearing what you have to say today in your talk. So let's welcome István.
Thank you so much, Danny, for the invitation and for coming, right? I guess you must like complex systems, and what could be more complex than the brain? So that's the topic for today. And I'm going to approach it as a physicsy system, using tools from physics and network science. And in the first part, I'm going to think about just the anatomy of the brain, what we know about that, and why is it interesting? Is it really a complex system in terms of physics? And then in the second part, I'm going to try to connect it a bit back to biology and ask questions about, okay, we know how the brain looks like. Do we understand why? Can we figure out the rules from the data that we have? And there are lots of open challenges in both space. And so to get started, right, we all like to think that the human brain is very complex. We have roughly 100 billion neurons, right? Nonlinear elements connected in a complicated way, roughly by 100,000 miles of connections, right, in each of our brains. And so it looks like an amazing level of complexity, but in just terms of the sheer numbers, maybe we are already obsolete because you can buy chips that have more transistors than the number of neurons in your brain. So maybe it is coming, right? That we shouldn't be that proud of our numbers, or maybe we should look into different numbers, right? Because for sure, even if these numbers are not yet there to beat humans in some tasks, it's getting there. Computers are improving. So maybe it's not about the number of units, maybe it's the network problem, maybe it's about the connections, right? And the typical human neuron has around 10,000 connections to other neurons, while a typical transistor has 3 connections to other elements, right? So we might beat silicon-based architectures for now, right? Because of the network aspect. And maybe it's not just about the numbers, but maybe it's about how those elements are wired together, right? And okay, let's see if I can. And okay, this was not a fair comparison, but if you are thinking about a more fair comparison between neuromorphic computing, those numbers are already getting there as well. It's not that far off, right? From where we are in biology, maybe other organisms, right? Not yet humans. And this is just individual chips, right? If you put many of them into a server room, you can get much more computing power and memory and all that. So all right, so maybe the brain is not as complex as we used to believe compared to these numbers, but let's try to understand it based on data. And I'm going to focus on 3 data sets here. These are all recent data sets. You can see the references down there. So not too long ago, half of the fruit fly brain has been mapped out. And since then, there have been recent developments mapping out the entire brain. I will talk about that a bit later. We also have a roughly a millimeter scale reconstruction of the mouse brain. And we also have similar data set in the human brain. So these are really amazing data sets. And one thing I want to point out here is that these are like 3D images of the brain, right? And the resolution is around 4 to 8 nanometers big. So each 3D voxel is roughly of this size, linear size, meaning that you see a lot of details. You can see individual synapses, details of the neurons, axons, dendrites, a lot of biology is hidden there. We are talking about petabytes of data, right? That is readily available. You all have access to these data sets. It's freely available online. You can play around with that after the talk. And there's more data coming. There have been already okay, this is slow, but there have been already similar data sets in smaller organisms that don't have this scale, but they're kind of complete brains mapped out already in a increasing list of organisms, and there is work in progress. Amongst these, some of them are really ambitious, like mapping out the entire mouse brain, right? So imagine having that, right? You wouldn't be able to download it to your laptop, but it would be an amazing data set, right? And as I mentioned, just last year, right, a few months ago, the entire fly brain has been mapped out. It's around 140,000 neurons with all the chemical synapses between them, presumably, right? Maybe it's not perfect, but it's pretty amazing. I'm not going to talk about all of these data sets, just those 3 that I mentioned. And but before that, I want to put the data into some perspective. So how big is this data set, right? So it's comparable to astronomical databases out there, like the Sloan Digital Sky Survey. It's bigger than all books ever written, right? Okay, AI is going to write more, so that's going to be changing. And it's comparable to what CERN is capable of producing and storing, right? And this human data set we are talking about, roughly a cubic millimeter, is just, a tiny percentage of the entire brain, which is going to be a million times bigger, right? If we ever get there, or the mouse brain is going to be much, much bigger than what we have now. So imagine mapping it out. That would mean storing millions of petabytes of information. And we don't just want to store it. We would like to understand why it is wired the way how it is wired. And some of that must come from genetics, right? And if you look into the human genome, you will quickly realize that it is just 3 gigabytes. So if you try to encode all the bits in the brain that we can map out eventually in the genome, it is not possible, right? So there must be some compression. And current estimates say that maybe it's just roughly about 100 megabytes of information that's encoding the human brain. But we don't know how and which piece of information. It would be amazing to figure that out, right? So that's the goal. We are not yet there, but that's motivating the story that probably not all details are important, right? So your brain is different from my brain, yet we can both perform similar tasks at a similar level, right? So maybe not all the details are important for most questions that you can ask about the brain. And from a physics perspective, again, we call a complex system if there is a simple way to come up with predictive models, right? Simple but smart way. If there's no such a way, we call them complicated systems, not complex systems, right? In physics. So hopefully the brain, some aspects of the brain are really complex, right? And to give you a first introduction to the data, we are talking about electron microscopic images. So the brain has been sliced up in these organisms, very thin slices, and then with electron microscopes it has been mapped out, imaged with high resolution. And here is some small part where you see some axons and dendrites, and then that is just a very small part of a much bigger image. And in each 2D cross-section or image, you see the same neuron coming up on multiple locations. So here are highlighted some of them. So if you think about it in 3D, with a 2D plane, with one image, you cross each of them maybe hundreds of times, right? And it's hard to realize in 2D which one is which. So you need to reconstruct the data in 3D. And that has been done using machine learning. That's called segmentation. So now we know pretty much for every location in these data sets of which neuron it is. And we also know if it's an axon or dendrite. And we also see synapses. And if you don't believe me, I want to show you a video about this, if I can run it. So let's see. So first what you are going to see here is the human data set. So this belongs, the brain sample belongs to a 45-year-old female patient who lives in Massachusetts and who is probably missing this part of her brain, but it had to be removed for surgical reasons. And we are zooming into the data, starting at a millimeter scale, and all the way into the highest resolution, where each pixel is going to be a few nanometers, right? So this is just one 2D slice. So the entire picture would take 600 screens this way and 400 screens that way, right? So that's how big the image is. And you have a lot of slices like that. So we're going through different Z coordinates here, different slices, and you see some artifacts. There are some issues with the data. That's why you need machine learning to process it. But eventually, it's really high quality or good quality enough so that you can reconstruct neurons in 3D space. And that's the last part of the video, hopefully. And this is how it looks like in your browser if you want to play around with this petabyte of information. On the left, you can see a 2D cross-section. You can click somewhere and that neuron pops up on the right? So that's one way to play around with the data. And they really fill the volume, right? Of course, there are some blood vessels and a few other cell types, but eventually neurons comprise most of the volume, right? So, and it's similar in all other organisms. This is actually the human data set, all right? So let me move on to the next slide, if I can. All right, so let's just try to see a single neuron. So that's the blue guy here, and what you see in different colors is just a tiny segment of its 4,000 partners. So you can see all those other neurons are forming synapses with this guy, right? So this is just one neuron in our brain, maybe a typical one, all right, at a millimeter scale, and you can zoom in all the way so that you can see individual neurons, right? So one pyramidal neuron. All right, so that's the data set, just to give you a sense. And let me try to move on. All right, so as I said before, you can actually see and train machine learning algorithms to recognize all synapses, and you can even classify them as excitatory or inhibitory. So you really have a lot of details about every single object in this space. And in principle, you have enough information to already run dynamics on top of that, right? You know enough about the biology, and people are already doing that. I'm not going to talk about that today. What I'm going to focus on instead is trying to understand this amazing level of complexity, like are all the details really important? Is the brain a complex system where some aspects are more important for modeling, predictive modeling, or is it really a complicated system, meaning that all the details are important? And if there are biologists in the room, they might think that all the details are important. Since I'm a physicist, I'm going to argue for the other story here. And we set out to explore this with Helen, who was a postdoc in my group, and we asked this question, okay, if this is really complex, can we use something from statistical physics to go after it, right? In statistical physics, we know that at phase transitions, at least, you see complexity. And we have a lot of theories and tools, computational tools to explore that, quantify that, and it can lead to interesting predictions and insights, right? So that's what we wanted to figure out here. And what we did here was very, very simple-minded. We just realized that neurons look like self-similar fractal-like objects, so maybe there is more to it, right? This is one signature that you see at the phase transition. If you think about a critical system, right on the boundary between two macroscopic phases, you often see this kind of fractal patterns emerging. That's not the only reason why these can be there. So we were wondering if that could be the case in the brain. And just to give you a simple model that can do this, right, you can just think about percolation. You just build a random labyrinth, you pick a grid, with some probability you fill in the sites, and if you put in enough, then you have a macroscopic connected cluster. If you don't put in enough, then you have small clusters and you can walk along between them, right? You can cross the system without running into a wall. If you are doing it with the critical probability, you see fractal-like patterns. So that's probably the simplest model that can produce these fractal-like patterns. And we were wondering if the shape of the neurons could come out of something like this, right? I mean, of course, it is not going to be described by this simple model, but could it be that it is not just that neurons are fractal-like, but there is more to it, that other signatures of criticality could be also there? Because there are also a large number of other kind of systems where the fractality is not related to criticality. So how do you do this, right? Okay, maybe you haven't done this before in physics, so there are a few things that we usually look into. We study different characteristics of the geometry of these clusters. In this case, those are neurons, so each object here, like the green object, would be a neuron. In physics, it would be a magnetic domain of a superconductor or a magnet, Ising model, you name it, or percolation clusters. It doesn't matter where it's coming from. You have complicated looking objects in 3D space, and you can study different properties. One of them is you can check in a given observation volume how big the biggest cluster is that you see. So how big is the biggest chunk of a neuron that you can see in a window of a given size. If there is fractality, this should scale in a non-trivial way with the linear size. It shouldn't go with the third power, as in 3D. These are not spheres. It should go with a weird exponent, and that's the fractal dimension. Maybe that's the simplest way to measure the fractal dimension. There are many other ways to measure fractal dimensions. I'm just trying to give you one example here. You can also look into pair correlation functions, and what it means is that if you are somewhere in the brain and you go some distance away, what is the chance that you see the same neuron there, right? And since neurons have these long axons and dendrites, you think that maybe it is relatively likely that even far away you can run into the same neuron. So what happens, we don't know what happens in the brain, we didn't know what happens in the brain, but if you think about it in critical physical systems, you often see a power law. So those are nice, we like them, with some characteristic exponent. So that's one thing that we can check for, right, in the brain. And you can also see, all right, we talked about the largest cluster in some volume, but you can consider all the different fragments of neurons in a volume, and they might give you some distribution, right, of how big they are. And you can look into a probability distribution. And again, in critical systems, that should be a broad distribution characterized by a power law, right? So, and there are many more measurements that people do in critical systems, and we always, or almost always, expect to see power laws, right? Most critical systems. So that's one thing that we wanted to do in the brain. So we started measuring things like these, and surprise, surprise, fractal dimension really works. Of course, in the right limits. So you can estimate with this trick that I mentioned, just measuring some observation windows and seeing the largest chunks of neurons, that the fractal dimension is around 1.6 in all three organisms. So that was surprising, because fly neurons don't look like mouse neurons or human neurons. They're very, very different, seemingly. Yet the fractal dimension seems to be similar. And of course, there are other ways to measure fractal dimensions. We also did box counting, if you know what I mean, but I'm not going to show that because you get consistent results. And by the way, you can do this for critical systems like percolation. You get a completely different number. So the brain is different than percolation. We knew that, but we see the same quantitative scaling. And here's an example of how neurons look like in the fly brain. So actually, we know that different neurons have different fractal dimensions. So what I mean by the fractal dimension that I showed you is kind of an aggregate statistics. If you consider all neurons together, the entire scaling in a volume, then there is a number with an error bar that you can assign to that. And there are interesting details here. For example, one thing is that we ran into, I'm not going to talk about many of these details, but one of them is that if you have a larger neuron, it has a larger fractal dimension. We don't understand why. If you have any ideas, you should write a paper. So we just got this data recently, and it's very interesting. We didn't expect that to be the case. At a critical system in physics, that wouldn't be the case, right? Small clusters would have the same fractal dimension as large clusters. Here, there is something more happening. So maybe the brain is even more complex than complex systems in physics. I would be happy with that conclusion. But before running there, let's see other observables. So one thing I mentioned that is really a hallmark of criticality is that the pair correlation function, or these correlations decay, has a power law. And that's kind of what you see here. It's not perfect straight lines, but what you see here is that all three brain data sets in three different colors highlighted have roughly the same slope. It's not an easy job to extrapolate slopes from not straight lines, but you can do finite size scaling. That's a well-known technique from physics. It gives you robust estimates. So that's what we used in all these cases. And again, surprising thing is that all three organisms give you the same number, around 2 point something. And again, it's very different from percolation, but there seems to be some higher than random chance that you run into the same neuron even far away. It doesn't decay exponentially. It is much more broad than that. So that's a strong signature of criticality again. And you can do many other things. In physics, we often look into notions of susceptibility, if you heard about those things. And in a clustered system, that just means the mean cluster size per site. So that's something you can again calculate, and at large enough scales, which is around the scale of the data, right? You can see that these measurements run parallel to each other. Again, you see the same exponent, right? So it looks like everything that we expect to look like a power law is consistent with the assumption that you see a power law. Of course, we only have that much data, only a cubic millimeter, but it starts from nanometers, right? You have many, many scales. It's not too bad. And the big surprise so far has been that the individual critical exponents in these three data sets seem to be consistent with each other, which is a big surprise, because if you look into raw numbers, like how big are the neurons, how many partners do they have, other things, those are very, very different, right? So, and there are more measurements, like, okay, I already mentioned to you one of these, like if I pick some part of the brain of the data set that I have a smaller part, like 0.1 millimeter across, like you will see a broad range of volumes of neurons intersected by that, right? Some of them are just passing very quickly, some of them spend more volume in there, right? And this is characterized again by a nice power law looking function with a critical exponent that seems to be consistent with what you would expect from physics. So this is the first non-trivial thing I'm showing you, maybe. Okay, everything that went beyond fractality was new, but here there is something much deeper, that it's not just that you can measure infinitely many things and you see infinitely many power laws with different exponents, but these exponents shouldn't be independent. That's what physics tells us. If you really have a phase transition, yes?
For anyone online. Yeah. So maybe I just missed this. This is just a clarifying question. Is the cluster size distribution, how do you define a cluster? I guess from your images and videos, it seemed to me that everything was clustered in the brain.
Yes,
it was one giant
so each neuron is a cluster, right? And then what we do here is a trick that we also do in physics, that you have some infinitely large system, you just take some box of that, right? And in that, you know, you will not see the entire neuron, you will just see parts of that, right? And okay, neurons might have a typical volume, right? But independently from that, when you just sample the data, right, in smaller boxes, you expect a broad distribution at a phase transition. And that was somewhat surprising that this happens in the brain, right? Especially if you think that neurons should have, at least each cell type should have maybe some characteristic volume, right? Then you would not expect a broad distribution. So maybe this is coming from the fact that there are so many different cell types that overall you see a broad distribution. Okay, I'm not making a statement about that because we haven't really checked it carefully. First of all, not all the cell types have been characterized in the data, so all right, so we are not yet there, but yeah, so this is just really what I said, that you're just subsampling the data and you see chunks of it that belong to the same object, same neuron. And I don't know, this doesn't have direct biological meaning maybe, but it's one of the things that we do in physics. And it provided one piece of insight for us, that the exponent that you see is related to the other exponent. So you can measure many different exponents, and we know that at a phase transition there should be only a handful of them that are independent, at most 3 of them typically, right? So if you measure 3 things, then you can predict all the other critical exponents for all the other quantities that you would measure, at least for those quantities that we usually measure in physics. And here is an example, and this relationship is highly non-trivial, right? So, and it seems to work. So that's just one example, that it's not just that it's a random coincidence that you see a lot of power laws, a lot of exponents, but these are also satisfying relationship between them. These are not independent. So really, as a summary of how far we got, right? So we see that that each of these data sets displays some signatures of criticality, the usual ones that you would expect. And one big surprise is that these exponents are consistent across different organisms, right? So that might indicate universality, right? That there could be something shared across these different data sets that is not apparent, right? When just look into them, they look very different. But in terms of these numbers, they are the same. And an even deeper evidence for criticality is that these exponents are not independent. And I showed you one example already, but there are other relationships between these, like eta is related to gamma over nu, things like that. So and these are all seem to work. They seem to work, right? So that's that has more convincing power for a physicist, I think. And but why are we doing this, right? And one of the goals of doing these measurements is to learn something interesting about the brain that you might want to reproduce in a generative model. Like imagine that your job is to sit down and come up with a computer simulation that could create a video like what I showed you, like a fake video of a statistical model that matches some key properties of the brain. So that's one of the goals here. So by measuring these universal numbers that are consistent across the different organisms, right, you could try to come up with a model in physics that can reproduce those numbers, and that could serve as a brain model, right, in some sense. Maybe it wouldn't be telling you anything about the developmental process of how the brain is built in biology, but it could give you some description, statistical description, predictions about the adult brain, right? So that's the hope. And so maybe you don't even need a new model. So maybe something out there is hiding that already matches these numbers. So that's what we wanted to check here. And if you look into just two exponents, fractal dimension versus the eta exponent that is related to the 2-point correlation functions, you see that the data sets are somewhere here, the brain data sets. And if you go through a bunch of different models in 3D in physics that we know, they typically are found on this line, and the brain is far off from that line. So two things are off that line that we know in physics. Both of them are disordered Ising models related to, so it is related to recent Nobel Prizes. So one of them is the spin glasses related to Giorgio Paris's Nobel Prizes. That's outside of this line. Another one is called the random field Ising model, and that's again related to many things, quantum computing and many other aspects, machine learning. And surprisingly, this random Ising model seems to have exponents that are very, very close to what we see in the brain. Again, this is a hard model to study, so we don't know precisely the exponents. The brain data sets have a limited scope as of now, so we have some error bars for those exponents, but they seem to be very close. And there's one more thing on this figure that I wanted to highlight before moving on, that there's one more structure out there that seems to overlap with what we see in the brain, and those are galaxy clusters. If you look at the sky, galaxies also cluster in a fractal-like manner. You can measure the fractal dimension. You can measure 2-point correlation functions in those structures. And those two numbers are in this regime, and they overlap with what we see in the brain. The complication is that in the case of galaxy clusters, the fractal dimension depends on the distance, on the redshift, right? If you go further, you see a different fractal dimension because the universe is aging, right? But at the right age, it looks very, very similar to the brain, right? In terms of these universal numbers. And all right, so there are still many open questions. This is just the beginning of exploring what physics can tell us about a complex system like the brain. So unfortunately, there are some of these that are really hard, like, okay, what is the control parameter? When you have a phase transition, you want to be able to bring the system across that phase transition. I don't know how to do that. I only have one snapshot of the human brain, but eventually we'll get more data. Maybe we will see something about this and learn what could be a control parameter. Then again, can we fully classify this, right? What else should we measure, right, to really get a deeper understanding? Probably this is already enough to some extent. And really the big question, can we generate something that looks like the brain, right? That's what we are working on. And maybe the random field Ising model is already that. The issue with that model is that no one ever tried to find neurons in that model. So they might be hiding there. Somehow we should just find them, all right? So that's a project we are working on. And on the more biological side, right, so why is it critical, right? Why do you need this level of complexity in the brain structure? And you might have heard about criticality in the brain, but that's always about the dynamics. And when people try to model those, they think about a lattice or a fully connected network. They don't think about a critical hardware architecture that runs the dynamics on top of that appears to be somewhat critical, right? So we think that this should be an important aspect of understanding brain dynamics. We are not yet there to connect the two, unfortunately. And of course, there should be some trade-off between cost and efficiency in the brain and so on. So there are so many open questions that we are just at the beginning of the journey, but there's more and more data coming, and maybe some of our observations will hold even when you go to other data sets. So this was the maybe most solid part of the work that we've done. But I know that some of you are interested in networks. I also want to tell you where we are with our understanding of the network aspects, because so far I did not talk about which neurons are connected and how, and you can look into the topology. of the brain. So far we just considered this data set as a material kind of, right? It's a 3D object, fractal-like domains. So what can we tell about that? You could separately study the network aspects, but in reality it is a spatial network, right? Neurons have locations. It probably matters how far another neuron is for wiring and other aspects, right, function. So really we need some spatial network models, and that's what we did with Anastasia, who is sitting here in the audience. So if you have any questions about the details of this work, she is probably happy to address those. So here the question was, okay, we know a lot of complex systems that come with nice complex networks, and we have models for those, right? Do any of those match what we see in the brain? Is the brain a scale-free network? Can you say something like that about it? Like in what sense is it complex? And can we again use tools from statistical physics to get a better sense of how the brain looks like, and maybe towards generative models, right? So that we can predict something. That's always nice to start with some data and make predictions. So this paper just got accepted few weeks ago, so it's out there. You should look into the details. I'm not going to go through all the figures, but I just want to show you how the brain networks really look like. So what we do is that for each neuron, you define a position. That's kind of the center of mass for now. It's a bit more complicated, but you can think about the center of mass. And then for each pair of neurons, you can check if they are connected. So here we are, Anastasia, we created this figure in the fly, mouse and human brains, showing you the entire data set. So this is how it looks like in 3D, kind of. So it's a mess, but there must be some complexity to it, right? For example, one thing you might notice that in the human brain, you see sparser and more dense layers. So those are really the cortical layers, right? And there could be interesting network aspects of those. So one thing that I have to tell you before getting to our conclusions is that it is not just that we have a synaptic network here, but you have the data to construct a contact network. So you can tell who the neighbors of each neuron are, right? And we are the first ones who are trying to argue that this is important, because even if two neurons would love to connect, if they're far away, they won't connect, right? So if they have matching genetics, it would be beneficial. If they're too far, they cannot connect. But on the other side, just because you have a neighbor, you're not always friends with that neighbor, right? So there might be more to forming synapses than just being neighbors, right? Okay.
Does it ever happen that two neurons are multiply connected? More than one synapse?
Sure, yes. And in different organisms, the level of that is different. So you can have multiple synapses. So that's the synaptic weight in some form, and it can be a scale-free power law in some organisms, right? Okay, according to the data. We are not really looking into that. We just ask, is there a connection or not, right? We wanted to get started, just basic topology, right? Together with some spatial information. But what I wanted to highlight is that probably it doesn't really make sense to just study the synaptic network on its own. What makes more sense is to first map out which neurons had a chance to form a synapse. That's the physical contact network. And on top of that, you can ask, okay, why do some of these become friends while others are not friends, right? So that's kind of the way how we formulated the questions here. And on the right, I'm showing some neuron examples that if you think about the soma, the center of the cell body of a neuron, it's not always in the middle. So the location that we actually use is kind of in the middle. It's kind of a center of mass or mesh of the surface of the neuron. And there is not much of a difference in most organisms, but in the fly brain, there's a big difference. So that's why it's important. In the fly brain, all the cell bodies are on the surface roughly, and then everything happens in the middle of the brain. So the location of the cell body is not really interesting, right? It doesn't seem to be informative, right? But what is really important is kind of where the center of mass is of a neuron. So that's why we use this consistently across all these data sets, just a technical detail. And to give you a sense of what we did here, we asked questions, okay, what is important about this data? For example, is a degree an important thing, right? How does the degree distribution look like? Is it enough to match it in a model to describe the data? Probably not. And if you need to go beyond that, what else do you need to include? And you could include physical contact information, that's C, degree is K. You can include some information about total wiring length. since it is so big, I told you it's over 100,000 miles in the human brain, maybe the brain is optimized for it, right? So that you don't waste the connections. You can use information about distance, right? And you can combine these things. So it was a lot of work to explore all these different models. And here are some key findings. Without getting into all those plots, you should really check out the paper. I have extra slides about it if you have questions, but this is what we learned, right? As a quick summary. Maybe it's enough at that level for this audience that no, it's not a scale-free network, but it's a broad degree distribution. So there are neurons out there, like in the fly brain, there is a neuron in the data that is connected to every third other neuron, like 1/3 of the data, right? So they're big hubs, but it's not a scale-free network, right? It is broad, you can quantify it, so it's not enough to think about it as a random network. It's somewhere in between random networks and scale-free networks. The distance dependence is exponential in these variables, so if you go far away, it decays very quickly, the probability that you connect to another guy. This has been well known in neuroscience at a much larger scale between brain regions. But here we are talking about neuron level scales, right? Sub-millimeter. And it's already true here. Interestingly, the length scale at which the connections decay is not the same in these data sets than between the regions. So there are lots of interesting details. Another thing that I wanted to point out is that if you can match with a model, like a maximum entropy model, the degree sequence, how many partners each neuron has, it gives you completely wrong distance dependence, because then you will also connect to things that are far away, so that doesn't work. If you try to match the distance dependence, exponential rule, you don't get the broad degree distribution, right? So these are somewhat in a conflict with each other. So what you need to do is you need to include both of these. You need to include the number of partners a neuron has and some information about their location or some notion of distance, right? And that's how you can, and if you include those two things in some form, then it's already a very powerful generative model. So that's the conclusion of this work, that you can include the degree of each neuron and the total wiring length, or the degree plus the physical contact information. And those two, either of those models are very good already. And if you start with those, they have deep predictive power. For example, I can just use those to predict synaptic weights, like what Rosemary was asking about. How many connections are there actually between these? So the input data, in the input data, we ignore that information. It's just topology. But then in our models, right, for each pair, you assign a probability that they connect. And that seems to be correlated with the weight. So we can predict the weight, right, without using that with these simple models, just using degree and some location information. And there is much more. If you just know the physical context, you can predict where the synapses are. It's not perfect, but it's better than random. It's not a random subset. And you can predict higher order network patterns, number of triangles, squares, and global properties of this brain data set, like effective resistance, transitivity, many other things. And in the human brain, as I showed you, there are cortical layers, so you can even predict from these models the level of anisotropy, that in different directions the brain looks different. All right, so already with simple models we can go very far, so you really don't need to capture all that information in a predictive model, and eventually it would be really nice to connect these two things, like the complex material aspect, that neurons are fractal-like. Somehow that should end up giving you the physical contact network. And then from the physical contact network, you can go to the actual synaptic network by using genetics, right, or wiring rules, because eventually neurons form a connection for functional reasons, and that is most likely encoded in some genetics, at least partially, right? So if you know which gene is expressed in which neuron, you could in principle learn about those rules. So that's the last thing I want to talk about. And here, we are not yet there to do this in the human brain or in the mouse brain or not even in the fly brain. We just don't have all the data collected in a standardized way. So we started in the C. elegans, which is a tiny worm. It has roughly 300 neurons. The brain has been mapped out before I was born, so we know about this a lot. And there are still many interesting open questions, and that's something, again, that we've been working on, and we really hope to get there in these other data sets as well. We have the entire fly brain. People are so good at mapping out gene expression nowadays that soon enough we should have the data to combine the two and figure out why each pair of neurons is connecting, right? And unfortunately, I need to tell you a bit of biology before we can get to the results. So there are two major types of synapses in the brain. You can have the chemical synapses that you might remember, but there are also electric synapses in the brain. And those are just some protein channels, right? So, and those are very different from chemical synapses, and their number is roughly the same as the number of chemical synapses, right? But they're much more simple because as far as we understand their biology, there are only roughly about 20 genes in the worm that encode how these are formed. While for chemical synapses, your guess is as good as mine. Probably all 20,000 genes play some role, right? Or at least thousands, right? It's very complicated. All right, so this is how the worm brain looks like in terms of just the electric synapses, and these are these protein channels. And so we know which two neurons are connected. We also know their gene expression. It's not perfect, but we have a good enough understanding of which gene is expressed in which out of these 20 innexin, or about 20 innexin genes that are important. And the idea is that if you have some neurons that express one of these innexins, some neurons that express the other one, and those two genes can form these channels, then you should get a bipartite network like this, right? Or these blue neurons should connect to all the white neurons. If you have some rule that, if you have a matching language, matching surface protein, we can just connect, then you expect to see clicks in the brain, right? So in the generative direction, if someone would give me the gene expression, the rules, I could tell you which neurons pairs have a chance to connect. The problem is that no one's giving you that data. We have the opposite, the inverse problem, that we know which are connected. We don't know anything about the rules. So can we figure out the rules? And this is what we could come up with in the worm brain. Looking into all these data sets, we came up with these 19 significant rules computationally using the brain. Gene expression, we predicted these microscopic rules that, hey, innexin 2 should connect to innexin 10. So if you have two neurons that express these, there's a 66% chance that they form a synapse, right? So this is what we did. And we are still waiting for the experimental confirmations. There is some evidence that this is actually correct. The real story is much more complicated. There are combinatorial rules, but I'm only going to talk about that if you have questions. If you want to see some formulas, let me just say that this is what we did. It's kind of a linear model that you can encode the gene expression table as a matrix. You can encode the connectome, the adjacency matrix as a matrix. And then after some thinking, you can connect them together with a wiring rule network, meaning that if one neuron is expressing a specific gene that has a binding rule with another gene that is expressed in another neuron, then there should be a synapse, right? So it is just some kind of matrix multiplication mathematics. The real problem is nonlinear. We are working on that. But if you just believe that a linear formulation has some merit, then you can solve this problem for the rules. So that's missing. We know the brain, we know the gene expression, we don't know the rules. And you can use linear algebra to find a solution. Unfortunately, it's not that simple. If you deal with complex systems, you know that there are always issues. Like the data is noisy, you need to regularize. You need to do statistical significance tests. And as I mentioned, just because two neurons have matching genetics and rules, it doesn't mean that they connect. You need to take into account the physical context, right? So who are your neighbor? Because you can only choose friends from your neighbor list, right? So if you include all of those, that's how you get those predictions, all right? And that's, again, I already had the citation up there. That's the work we published. And you can do similar things with other data sets. We've done baby steps, and I'm almost done with my slides, so to leave time for questions. So you can, for example, think about polarity, like it's very hard to understand chemical synapse formation. As I told you, we are still working on that. But once you know that two neurons form a synapse, the fact whether they are forming an exciter or inhibitory synapse only depends on a few genes, only 3 neurotransmitters and a bunch of receptors. And we know a lot about this. So we went after this data set and asked, okay, knowing what we know, can we make predictions that can be validated? And that was done with the help of Thomas, who is also sitting here in the audience. So, and again, this is kind of an overview slide about what you can do in this framework, right? Depending on the input data that you have, you can go after different questions. You might ask, what if I have gene expression and rules? Can I predict the brain? I told you that's simple. That's problem 3. A bit harder problem is I give you gene expression, but no rules. But I tell you something about the brain. So first you need to infer the rules, and then you can make predictions. And the even harder problem is just the network aspect. I don't give you any knowledge about the genetics, no biology whatsoever, just give you a network and you make predictions, right? And surprisingly, you can even do that. Okay, so if you have questions about that, I'm happy to go after that. There is some details about the mathematics here. Don't look into the equations if you don't like those. I just want to tell you what really happens here. So in terms of polarity, it's really kind of a social balance problem that the enemy of my enemy is my friend. But it's a bit more complicated because you need to go one step further. What really happens is that, okay, for each of these examples, so this is all encoded in that mathematics. Let's look into one of these figures like this one. What does this mean? It means that if you have two neurons that have the same partners, and one of them connects them with positive links, the other one connects them with negative links, right? You can use this correlation to infer missing links, right, or polarities. So if I know that this is a pattern that is there in the brain, right, even if I don't know this link, I can predict it back, right? Because, hey, they must share the same partners, and this always goes with the opposite sign, so this should be negative, right? And all the other patterns also make sense in a similar way. It would take much longer to get into details, but again, there is a reference here that you can look up about the math. And for the physicists in the audience, let me just mention that there is even more to this story, because this mathematics, even though it looks simple, is just like how you calculate distance between points in a non-metric space. And that's what we do in special relativity theory. And so eventually the mathematics of space-time and special relativity theory comes up and you try to describe latent space, genetic space, and wiring rules in the brain. And that's a lot of fun. And if you think that these rules might change from the head to the tail or under some chemical influence or based on concentrations, that means that this space is not homogeneous, it's not the same everywhere, and that brings in the mathematics of general relativity theory. So there are connections here to deep mathematics, and maybe there was also a predictive power. I'm happy to talk more about this if you're interested. What does it mean to have space-time directions in genetic or space, or time-like directions in genetic space? I'm happy to tell you more about this. Just to tell you, okay, just to show you that this is actually useful for something, right? This is an ongoing work with Engin Özkan from University of Chicago as part of an ITMB collaboration. We are trying to do this for real for chemical synapses right now. We just put up a preprint where we did a joint work of an experiment where we tried to identify all the genes that are important in chemical synapse formation. And for the first time, we have a high-throughput data set that tells you which of these proteins connect to each other. And that should be very informative about how synapses are forming in the brain. So there are lots of follow-up papers coming out of this experimental data set. And on the left-hand side, what you can see that we can predict this as well using our framework. If you combine the brain data set, right, with the genetic information, transcriptomics, you can also predict the network just using computations, right? And as of now, there is no perfect match between the two. These networks don't look the same, but by improving the mathematics, I think we can really get there to show that you can get meaningful predictions. And already here are some examples that look very, very interesting biologically that have been found both in the experiments and in the computational framework. All right, so again, the story goes much further. So you could ask similar questions in other complex systems, like imagine, for example, I don't know, online dating, right? You can think about people connecting to each other. There must be some features involved, right? So just like neurons and their genes, could we understand the rules, right? So the same mathematical problems come up. So tools that are successful to understand those systems can be also used here and the other way around. And as a big picture overview, this is what I was trying to show you, that you can think about these data sets, really amazing data sets, in terms of just the geometry or anatomy. And we see hallmarks of criticality, self-similarity, and we can make predictive models of that aspect of the brain. Or you can use tools from network science, especially spatial networks, and then make predictive models about that, and maybe even connect back to deeper mathematics and physics. But unfortunately, what we are missing right now is really a connection between these two approaches, right? So we still need to work on that. So if I know the topology, can I argue that the anatomy should be complex? Or if I understand anatomy, can I argue how the topology looks like, right? So we are not yet there. But hopefully, some of you get excited enough about this that you can help us to get there. All right, with that, let me thank you all for your attention and see if you have questions.
Thanks, István.
Thank you, István. I have 2 questions. The first is about the relation between gene expression and the formation of synapses. The example you gave, you understand, was for C. elegans, where I don't think there is too much learning after birth, right? So you are given the network, and once the network exists, you can see whether there are correlations. I'm more curious about the inverse of that, whether you could predict the formation of new connections between neurons based on their gene expression prior to the formation of the connection.
So that's
Which of course would require going to another organism.
So that's a great question. I think you can do good enough predictions, right? I don't think that you can do a perfect job without understanding the function or the dynamics or things like that, right? Or the task, but you can do a good enough prediction. Probably one can predict if a synapse could form, right? And then it might be realized or not. But for that, you need the rules, right? And that's the problem, that we don't know the rules. So that's the catch-22, that if I knew the rules, I could predict which synapses can form. But since I don't know the rules, first I need to infer them from which synapses formed. So it's kind of a circular problem.
Oh.
But one can check it at multiple stages, right? If you set up an experiment, one can, ideally, one would want to map out the brain before training and after training, or in different developmental stages, and also get the matching genetic information. And people are working on this.
I was thinking that correlated spiking leads to the formation of and spiking and gene expression.
So in the mouse brain, for example, there's also a dynamical recording about these neurons for the very same brain that I showed you. We haven't yet looked into that, but we don't have the genetic information, right? So that's the issue.
Very quickly, I don't want to take too much time from others, but when you talk about criticality, what exactly you are defined as criticality? Because in physics, usually we associate this to a mechanism, right? When you're close to a phase transition, you have something that can be mechanistically described, and then as a consequence of that mechanism, you end up having some power loss. Right? The power loss is is the after fact result of it. But power loss also show up in many other places where you don't have anything to do with criticality. So here, I of course do see a relationship between cluster size in percolation and the size of neurons. But is there any mechanism there that you didn't talk about? Like, did evolution at some point decide to merge neurons and make big neurons out of small ones?
So I wish I knew. So yeah, great question. Again, we don't know the answer right now. So going into this project, we expected that maybe the fly brain is less complex than the human brain, right? And maybe you will see no power laws there, right? Just exponential decay. And then in the human brain, suddenly you see power laws with beautiful exponents. But then we looked into data, and you see the same thing in both data sets, right? So that was a big surprise. So if there something like what you mentioned happened, it must have happened before the fly brain, right? Or earlier stage of evolution, I think. And we need to look into further data. But people are generating more and more data like this. So, but again, going back to this, like, it's not just that we see power laws; those exponents are not independent, right? So that's something that you can't really easily explain by other theories out there. The other thing I wanted to mention is that what we are really saying is that there is a brain universality class, right? Just an anatomy universality class, that if you find a model in that universality class, it will be a good model of brains, right? Across multiple organisms. And that universality class doesn't only predict critical exponents, right? There are many other things that are universal, like amplitude ratios and bunch of other scaling functions, things like that. So there are infinitely many things that this framework could predict, right? If you can find a model that is close enough. So that's the goal here. And each model will come with its own mechanisms, right? But again, it's not yet quantitative related to our observations, but as a big picture idea, right? We all know what we're looking for, right? It must be a compromise between wiring costs, right, and complexity, right? You want to have a complex network, but with a minimal wiring cost, right, and place the neurons optimally, right? So somehow the trade-off between those two things, just like trade-off between two free energy contributions in physics, leads to this criticality, right? So that must be the thing. We just don't have it written out, right? And maybe we won't even get there ever, right? So, but to have predictive models, I don't need to know that. It's enough to know the universality class, right? Okay.
Other questions for István?
I was thinking about the relation between the exponents that you showed, the 3 minus, and I was wondering if you could give some intuition of how that comes about.
All right. It was I'm not sure if I easily can go back to that. All right, let's try to run back. So I showed you 2 examples. There are many more, right? So since there are only 3 independent exponents, and you can measure infinitely many, in principle, there are infinitely many relationships like this, right? So what this one comes from is that there's, okay, I can write it down. I'm not going to do the full derivation or something, right? But usually what you see is that this tau exponent that gives you the size distribution of these fragments of neurons that you see in some observation window, right? That exponent goes as 1 plus physical dimension over fractal dimension, right? So that's how it usually is introduced, right? And then this is a different form of that because there's also another relation that relates physical dimension to gamma over nu. Right? And then combining the 2, you can get this result. So it comes in an indirect form. So this is not the usual way how we write it out in physics, right? In physics, we have 2 separate formulations and we combine them. The reason why I'm not doing it that way is that this notion of dimension comes up if you try to do it that way. And you might assume that it's 3, right? That the brain is 3-dimensional, and you just plug in a 3, and it doesn't work. So that is known as this is a hyperscaling relation that breaks. So I wanted to get around that problem that not all of the relations are valid. Some of them are broken. So this is an example. And what it really means is that the brain is not 3-dimensional in this sense. You need to plug in an effective dimensionality. And that effective dimensionality is the same across these 3 different organisms. And in the end, it all works out. And maybe if I can just one last thing to add to this. Oops. All right, so this is just too slow. So if this relationship is valid, that the dimension is just 3, that means that the model is on this line. And the brain is off that line, and spin glasses are off that line, and the icing model with disorder is off that line because it breaks this specific relationship. But still, these exponents are related. You just need to leave out the dimension because you don't know what the real dimension of the brain is, right? So, okay, sorry.
Questions? I have a quick one that's related to this, which is the single neuron. Is there a fractal dimension you can evaluate for individual neurons? And is it dependent on neuron type, and does it relate to these material fractal dimensions?
So yes, I already showed examples. So you can do it for individual neurons. Again, some methodologies work better for this purpose than others, right? So in physics, there are so many different ways to measure fractal dimension. They all work. When you try to put it into biological context, some of them break. But there's a way to get individual neuron level fractal dimensions. And in the paper, we have a figure where we show how the fractal dimension is distributed across different cell types in the fly brain, because there we have cell type classification. And indeed, they are different, but the ranges overlap. So it is well centered around the statistical mean value that I shared, right? But just like here, right? That it's, I said that it's around 1.6 or something overall, but for very small neurons, it's actually different. And yes, So these are great questions. And if you really wanted to come up with a better model for the fly brain, you would need to take into account cell types, maybe model in each of them individually, but we are not yet there. Okay.
Show this interesting plot. Any other questions? If not, let's thank István again.
Thank you.